All right, thanks very much for your kind words, Fiona. And it has been an absolute pleasure working with you and the AWI Extension team. Now, just give me the thumbs up and tell me if you can see the screen, okay?
I've got your screen. Perfect. Thanks, Matt.
All right, lovely. Well, we'll roll on into the presentation, and we do want to leave time for lots of questions at the end, so I'm going to roll through. The first thing is the season, and we're in spring now, and the unique thing about spring is that we consistently have moisture in the soil in spring throughout New South Wales, and that's something that we can't count on at other times of the year. But because we've come through the cooler part of the year when we've had more cloud cover, and we have been generally, lower temperatures and less drying off, we go into spring with higher soil moisture. Looking at the past 3 months, we've had, average sort of rain over the end of winter. And that's been greater in some areas. And as Fiona mentioned, 40 mils of rain in Armidale this morning means it will be topping up the soil moisture. Looking ahead to the season, so September through to November, the BOM is telling us that the next 3 months are going to be a bit above average for spring rain. So that's good for worms. That's actually giving us more moisture at the soil surface where the worms live. The other thing that the BOM tells us is the next 3 months are going to be warmer minimums than normal. And that means the worms will be more active in their development and more active in hitching a ride on the grass so that they can get up to places where the sheep like to eat. Now, for a little bit more insight, we do go to Farming Forecaster from Local Land Services. And this is a really handy tool to use to get specific information about a particular area. And it's been very valuable for our clients down in the Southern Tablelands, so members of Tablelands farming systems. And they've made great use of this. These particular examples come from the Hunter Valley. This is from the Upper Hunter at Merriwar. And we can see that when this was taken, due to the winds and a bit of drying off, the top 10 centimetres of soil is decreasing in moisture. And in fact, that's a bit counter-cyclical, from what's normal at this time of year. But it also gives you an indication of the amount of herbage available now. And that's pretty tidy. It's about 2,000 kilograms of dry matter per hectare, and projected what's going to happen into the future. So looking at the combination of these figures and the BOM, we can say that there's going to be reasonable pasture cover well into summer in the Merriwar region. It's not always the guaranteed though, because you look at the modelled growth and it's only looking at 12 kilograms per hectare per day. If we look at Singleton area, we get a similar sort of prediction, but the current growth is only 2 kilograms per hectare per day, and that will really benefit from some more spring rain. And then up to Wingham, which is generally a little bit wetter over the summer, and we can see that the projected pasture growth, it looks like it's set to decline, or it does in an average year, but the current growth is 49 kilograms per hectare per day. So these give us a bit of an indication of what to expect from the season, and a little bit later we'll correlate that with what it means for each of the worm species that are present in the different areas of New South Wales. The other thing that we really do need to always consider is the body condition score of our sheep. Now, if you've got scales and you're regularly weighing the sheep, then this is just an adjunct. But across the board, if you're using body condition score and recording it on your Lifetime U app, then that gives you a very good record of not only where the sheep are now, but where they're going to be if you apply a certain nutritional regime. So you can always improve their body condition score. For example, now, when there's a lot of sheep that are being prepared for joining, to get them into the right body condition score so that they will have A, high conception rate, and B, where you want it, to get them twinning as well. Now, if you haven't already done the Lifetime U management course, I'd strongly recommend it. It's probably the single best course you can do to upskill yourself on how to manage ewes right throughout the year to maximize their reproductive potential and also their health. So that's another AWI initiative, and you can go to the Lifetime U Management website and download the registration details for your area. Okay, getting back to worms. Now, there's been a lot of research done that shows that the humidity and the temperature has a big impact on worm development and survival. And so we know that for the black scour worm, for example, that this graph is based on, if you take them down to between, say, 10 degrees and 12 degrees, it takes 22 days for those larvae to develop from the egg. However, if you are in a warmer period where the temperatures are above 30 degrees, they will progress from egg to infective larvae in just 3 days. The other thing that is important is the percentage of eggs that hatch. So you can see at the peak in that zone between about 15 degrees and 25 degrees, the sweet spots there, which is temperatures that we often encounter in the springtime. We've got about 50% of the eggs will actually hatch and make it through to third stage larvae. So that's important because if we have very fast development but only a few eggs hatch, we are not going to get very contaminated paddocks. But this time of year, when we've got those very optimal temperatures, we have very good survival of the worms and high level of egg hatch. And then moving on to larval development, we've got 4 different graphs here for 4 different types of worms. The yellow one is the barber's pole worm, is the black scour worm that lives down in Victoria and Tasmania, Trichostrongylus vitrinus. It's a cold weather worm. It's suited to those environments. We don't see that much of it apart from some parts of southern New South Wales. The brown stomach worm, in contrast, has an ideal larval development temperature of 19.5 degrees. So round about this time of year is ideal conditions. And then the other 2 worms that we often encounter, which are the black scour worm that's commonly seen throughout northern and central New South Wales, Trichostrongylus colubriformis, as well as the barber's pole worm. They prefer temperatures up around 26, 27, and they have much better development rates at those temperatures. So as the temperatures get warmer, you'll see greater development of the warm weather worms, black scour worm and barber's pole worm, and less development of the cold weather worms, which are the brown stomach worm and the cold weather black scour worm. Now, what does that mean in practical terms? It means that, for example, in 2019, which was a fairly dry year in comparison, we had a lot of mixed infections throughout New South Wales. And these are figures that we just collated from our own laboratory here at Dorbert's. And if you've got a white circle, it means there's no barber's pole. A yellow circle means up to 30%. Orange circle means 30 to 70%. And then a red circle means greater than 70% barber's pole worm. We can see in that year, there were a lot of mixed infections. So a lot of yellow and orange circles. But in 2023, these are Shire averages for all the different places that we got samples from. They've almost all turned red. So we had a lot less worms from the other species, and barber's pole worm had taken over. Now, this is largely a result of the wet years that we had. So the 4 years of La Nina back to back meant that the barber's pole worm not only survived on pasture, they reinfected sheep in springtime and were multiplied inside the sheep because they lay so many eggs. Not really sure what's going to happen this year, but as we can see from the requirements for each of the different species of worms, we're fast moving into ideal conditions for barber's pole worm. Okay, well, let's look in a little bit more detail about the worm life cycle and see how we can use this to our advantage. So on average, the worm life cycle takes 21 days, and that's 21 days inside the animal, and it's pretty regular because it's a constant temperature, and the pH of each of the organs where the worms like to live is fairly tightly controlled. So it's almost like an incubator, and those worms have got very, very predictable conditions inside the animal. Now, despite us saying 21 days, there are a few exceptions. The nematodyrus worms, the ones with the great big eggs, the thin-necked intestinal worm, they can take as little as 14 days before they start laying eggs. And barber's pole worm can also be quite fast, down to about 18 days. On the grass, once they hatch out of the egg, they take minimum 3 to 4 days, but they can take up to 30 days to develop. And of course, the higher and the lower ends are associated with lower survival. So this time of year, when we've got temperatures right in that sweet spot, 15 to 25 degrees, is when we have the highest survival of egg through to infectious larvae, the L3 stage, and that's why we get very badly contaminated pastures very quickly. Looking again at this stage where the eggs reach the dung pat and they hatch inside the dung pat, this is critical for egg survival. So if the barber's pole worm are in the dung pat and it's very dry or very cold, they will die after only 5 days. And so if you have, less than your typical 2-day rainfall that activates the barber's pole worm in the dung pat, then they will just die away, and you do not get those eggs hatching into larvae and the larvae surviving. But the black scour worms and the small brown stomach worms last a lot longer. The black scour worms typically over 2 weeks, and the small brown stomach worms a month or more. And so they'll lie lurking. The eggs will lurk in the dung pat, just waiting for that shower of rain or the right temperature to come up, and then they'll emerge from the dung pat as infective larvae. Now, of course, for those eggs to develop into larvae and the larvae to survive and molt and turn into 2nd and 3rd stage larvae, we do need warmth, we need water, and we need oxygen. We often forget about the oxygen component, but for example, if you got all the dung and you put it into a compost pile, then you're likely to have not enough oxygen at the center there, and that's enough to sterilize those dung piles. And then the next stage, of course, egg to 3rd stage larvae, the maximum is about 55%. And on average, we're seeing about 1/4 to half of those eggs that are laid make it through to infective L3 or third stage larvae. And that does require, however, moisture levels between 40 and 80%, and the ideal moisture level is about 60%. Now we're talking about the humidity at the place where the worms are living. So for eggs that are in the dung pat, that is inside the actual dung pellets, and for larvae that have wriggled out, that's in the area around the top of the soil surface and in the first 1 to 2 centimeters of the grass sward. Okay, so we've covered the development of worms from the egg through the larva. Now we're looking at the worms on the pasture. What are these sheep looking at when they look down into the grass? They're not just seeing grass, they're actually seeing worms as well. So if you take out your magnifying glass and look at the pasture very closely, you can see a dewdrop, and inside the dewdrop there are dozens of worm larvae just waiting there for the sheep to come along. Each one is only about 0.7 millimeters long, so they're very difficult to see with the naked eye, and you need to actually use a magnifying glass or a microscope to be able to see them. And they're generally distributed down in the bottom couple of centimeters of soil. Now this is important because we know that sheep really do like to graze down low, and if you look at sheep, they typically put their head right down to the ground. They're not ripping off the sward like cattle do, or putting their feet up on a tree and eating the leaves out of the tree like goats do. They're grazing down low. So we have to take that into account. The sheep will pick up lots of worms because the highest concentration of worms is down where the sheep typically graze. Now, if we want to work out how many worms are on a pasture, we can actually do that by looking at the numbers. And we can say, just for example, we've got a typical 5-hectare paddock, and it's got 2,000 kilograms of dry matter per hectare. Then the feed on offer is 2,000, paddock size is 5. We measured the worm egg count of this mob of sheep, and it's 337 eggs per gram. And we know that on average, this age sheep produce about 1 kilogram of dung every day, so 1,000 grams. There's 100 head in the paddock. About half of the worms develop at these temperatures. They're on the paddock for 10 days, and about 3/4 of the larvae will survive those 10 days. So we can say that the number of actual larvae that are produced by this mob of sheep is 168.5 million, and the pasture contamination is that number divided by the amount of kilograms of dry matter and the area of the pasture. And so we can say that this paddock will have, or we can estimate, will have a contamination of 12,000 odd larvae per kilogram of dry matter. And so if we put lambs into this paddock and expect them to eat 1 kilogram of dry matter per day, then each day they're also going to eat 12,640 worm larvae. So that helps us to then work out how badly contaminated each paddock is and how we can use that to our advantage to avoid the badly contaminated paddocks for our most susceptible animals, which of course are our lambs and our pregnant ewes. The next question is, well, how long do the worms survive? And that's very easy to work out because the research has been done. This one is for barber's pole worm that shows that at high temperatures, they decline very quickly. In fact, 90% of them will decline after only a couple of weeks. And at low temperatures, for example, over winter, they decline very slowly. It might take 3 or 4 months before 90% of the numbers decline. Now, that's because when we look at the larvae at different temperatures, we can see that at high temperatures, they're very metabolically active. They move a lot. and at low temperatures, they're saving their energy, and that's why they're going to survive longer. So you can imagine over the winter, when typical temperatures are 5 degrees, the worms are hardly moving, serving their energy, they're going to survive a long time. And this particular graph comes from the Paraboss or WormBoss website, and it helps you to work out, how many weeks you need to spell a paddock for different temperatures for 90% of the worms to die. And of course, if you're starting off with 12,000 and 90% die, then you're left with only about 1,000 worm larvae per day. And healthy sheep should be able to cope with that type of worm burden. But if you put them into a paddock that's been spelled so that 90% die, but you started off with, for example, 50,000 larvae per kilogram of dry matter, then it's only come down to 5,000, and that's still a very high worm burden for particularly lambs to be picking up each day. Okay, other tools to reduce pasture worm contamination. One of the best ones is using cattle or dry stock on rotation to prepare the paddocks, and that's because the crossover of worms between cattle and sheep is very low, and generally you get a good pasture cleanup by using cattle to graze the pasture first. The other good thing is that the cattle tend to graze higher on the sward, so they will clean up a paddock that sheep won't be able to. And the second thing is, when you put the sheep into a cattle paddock, they will graze closer to the cow pats because the cattle have got an innate dislike of grazing within a metre of a cow pat. So the sheep will graze much closer to the cow pats. So you actually utilise more of the pasture as well. You can select for ASBVs, that's Australian sheep breeding values, of your rams, and that also applies to goats and machos if you're an alpaca producer as well. BarberVax is a very handy tool to vaccinate the sheep against Barber's pole worm and reduce the number of eggs that go onto the pasture. And so is biowormer, which is actually a fungus that's been packaged up in a feed that you feed the sheep, and it attacks the worm larvae and kills them in the dung pat before they have a chance to go onto the pasture. The only thing is, it's a bit expensive for very extensive places, mainly used on smaller places or very valuable animals. You can rotate your pastures with short grazings, and the idea is to get the sheep off the pasture before the worms have developed from when they pick them up off the pasture. And spelling the pasture for long periods, this may not be a good idea if you're running high stocking rates, because it may take pasture out of commission for a long period of time. Again, low stocking rates will also help with low worm pickup. Shorter joining periods are always beneficial because you have less time for the lambs to be in the lambing paddock, and also the younger lambs are much less likely to pick up worms than older lambs. So older lambs on a lambing paddock are always going to be very wormy. And similarly, scanning and drafting your multiples and singles and dry ewes is a huge benefit, because the dry ewes will have almost no worms or minimal worm burdens. Singles always increase during the pregnancy and lactation, and multiple bearing ewes have the highest worm burdens of all. Now, if you do lock up paddocks, you can actually produce very nice lambing paddocks, thanks to the natural attrition of the worms on the pasture, and that's particularly good if you sow a good feed for those ewes so that they can have maximum lactation and look after the lambs as well. Making hay or silage is always useful, because you're exposing the worms to extra UV by taking away the grass sward. You're actually packaging up a lot of the worms so that they die as well. Okay, well, let's get back to pasture larval counts. Now, this is a technique that we can use, just like the agronomists go out on pasture and take both plant samples and soil samples. We can take plant samples and submit them to the laboratory by weighing them, washing them, filtering them, and collecting the worm larvae. And then we count the larvae under the microscope, and you can see pictures there are from our lab where we had some very enthusiastic vet students who worked on a project for us, and they did pasture larval counts from all over the district. And when we looked at the counts, the highest ones from the larvae, the samples that they collected were about 40,000 larvae per kilogram of dry matter of pasture. And more typical was, between 10 and 20,000 larvae per kilogram of dry matter. And we know from other research that's been done, for example, from Invetus in Armidale, that getting to 20,000 larvae per kilogram of dry matter is not unusual in paddocks, typical sheep paddocks in New South Wales. However, getting back to our pasture larval counts, this is not offered by most laboratories because it's very time consuming, very expensive, and there is also a bit of an issue with the sampling. So if you sample using two different techniques, you might get two different results. So even though this is a great technique, it's not a practical one for using. We have to actually estimate what the pasture larval counts are using that previous technique, knowing the worm egg count of the mob of sheep, how long they were on there, and then estimating like that. And you can come up with a very simple system that allows you to put maybe a red dot on the lambing paddocks because we know they always get very wormy, or on any paddock that's had wormy sheep in it. And you can say, well, those sheep, we know they had a worm egg count of 800 eggs per gram, for example, and that was only 2 weeks ago, so that paddock stays red. And then as the contamination decreases, you can give them an orange tag, and only after you've done the necessary precautions for a low contamination paddock, for example, taking off hay, rotating with cattle, or spelling, one of those things or a combination, then you can give them a green light to allow you to put in your susceptible stock. And just coming back to ewes and lambs, we know that there's a phenomenon exhibited by all lambing ewes called periparturient relaxation of resistance. And it simply happens because ewes around the time of lambing and lactation prioritize the lamb and the milk production over their own health. And so the worm numbers rise and the worm egg counts rise. And so lambing paddocks are always highly contaminated. If we're able to use our microscopes and look in, we'd see there is a lot of worm larvae ready to infect those sheep. So to get around this and put sheep, particularly pregnant ewes that are about to lamb, onto uncontaminated paddocks, there is a technique called smart grazing, and you can actually develop your own based on the first principles. And this particular example was outlined by Justin Bailey, who, along with Steve Walkton-Brown and Lewis Kahn, did a big research project on this back a bit over 10 years ago. And they showed that ewes that were set stocked had very high worm egg counts. In fact, it didn't take them long to get up to the stage where they really did need drenching. However, ewes that were put onto paddocks where they'd been prepared by cattle or smart grazed with dry sheep, treated sheep, had low worm egg counts and didn't even need drenching. So there's some good case studies there from the Armidale area. And thanks to that, we've got smart grazing techniques that have been developed there, and also from the McKinnon project in University of Melbourne, that are available on the Wormboss website to download and use. Okay, next, let's look at flock management, and we just want to look at some of the ways you would observe sheep to know how wormy they are. Just the same way that our wool classes observe the fleece and the staple and work out, how to class different types of wool. We're doing the same thing with sheep, and we probably shortchange ourselves often. We're not using all of these tools in the toolbox, but post-mortem is actually a very handy method, especially for veterinarians. You can actually see some worms and others that are too small to see, such as the black scale worms, brown stomach worms. We can collect from the guts and put them under a microscope to see them and even count them. Processor feedback is invaluable, particularly given the new MLA My Feedback website. And I strongly encourage everyone to get onto that so that you can get really detailed feedback on both the carcasses and the health indices that you get back from the processors. Body condition scoring we've mentioned, and as well as body weight. Then we've got the Pharmachar score, fecal consistency scores, and DAG scores. So let's just go through some of those. This is a picture from the sheep's abomasum, and this is where the Barber's pole worms live. And so the Barber's pole worms are so big, you can see them writhing in the image there, and you can actually see them. However, what you can't see are the brown stomach worms and the black scale worms, which may be causing just as big a problem, but they're too small to actually stand out when you do a post-mortem. But at least you'll see the Barber's pole worms. However, if the sheep's already dead, then that's too bad. What you really want to do is to be able to predict when the worms are a problem before the sheep dies. And you can do this using the Pharmachar method. And this is a simple method, very reliable, of examining the lower eyelid of the sheep. And that's been quantified in this method called the Pharmachar system. And again, you can read more about this on the WormBoss website. And there's a chart that shows the gradation of colour in the lower eyelid from a nice red colour, where the percentage red blood cells in the bloodstream are above 28, down to a white colour, where it's estimated that the percentage of red blood cells, or the packed cell volume, is less than 12. And so you can examine the sheep and have a bit of a look and work out what colour they are, and that gives you an indication of how critical they are before they are going to succumb to Barber's pole worms. The other telltale sign of Barber's pole worms is bottled jaw, and you'll also see this with liver fluke. However, it's often a reliable sign that there's fluid accumulating, some level of blood loss, and that's a sign that those animals probably need treatment. Okay, now it doesn't just indicate that you need to treat the sheep, because if you come back a bit further in your helicopter view, you can actually say that you can stop the animals from getting into that state by feeding them better. So we know by selecting for genetics, by doing worm egg counts on multiple animals, submitting them for ASBV assessment, we can select sheep for resistance or immunity to worms. But by feeding our sheep, we can improve their resilience to worms, their ability to cope with the worms. And so this trial was done on 3 mobs of lambs that had been given 10,000 larvae of Barber's pole worms 2 days in a row. Now, the first mob of lambs had a growth rate of 75 grams per head per day, so a fairly low growth rate, and their nutrition was kicking in about 3% of their body weight. So they're given a ration about 3%. The next group had a 3.3% body weight ration, and they were kicking along at about 125 grams per head per day. And then the third group were doing 200 grams per head per day. Okay, so the impact of the barber's pole worms on the lambs that had the highest level of nutrition was pretty mild. Their worm egg counts went to 550, 925, and then actually went back down again by day 28 after infection. However, the impact of the lambs on the two lower growth rates was very high. In fact, in the lowest growth rate lambs, the worm egg count went straight to 2850, went up to 7,900 and stayed at above 7,000 right through to day 28 post-infection. So a very simple experiment, but it illustrates very clearly that lambs on a higher plane of nutrition are going to be more resilient against the worms and able to fight them off better. Okay, now getting back to genetics, there's a lot of effort being put into sheep genetics, and again, I'd refer you back to WormBoss to look at the details of breeding for worm resistance in your sheep. Okay, let's look at some of the drenches or treatments, and I just wanted to remind producers that Barbavax is available, and for example, a lot of sheep at the moment are being given Barbavax treatment at lamb marking, and then again 3 weeks later, and then again at weaning, so that sets them up for immunity against Barber's pole worms. And the Barbavax vaccine is being used very widely, not just in northern New South Wales, but all the way down to Gippsland and some parts of South Australia as well. And the flip side of it is you can actually set up your ewes for protection with Barbavax by injecting them prior to lambing as well. So now is a good time, particularly with the warm spring weather and the late winter warm spell that we've had, to start thinking about your Barbavax vaccine program. If you haven't already started, now is the time to do it. Second thing about treatments is that people often buy sheep and they're badly contaminated with worms and they bring on resistant worms. So just a reminder that a quarantine drench is important, and a single drench these days isn't enough to protect sheep when you actually buy them in, because there's so much resistance around. You really do need to drench them twice. So one of the new drenches, for example, StarTect or Zolvix, or the new active ingredients, and then follow it up with other active ingredients such as a combi drench, which contains a white and a levamisole, and make sure you get 4 active ingredients in. Now, if you want to monitor resistance around Australia or in your region, you can look at a couple of different websites. The first one is Sheep Tracks, which has been put together by Zoetis, and it's very useful for the number of trials that have been done. Now there's hundreds of trials being done and put into that website. And the second one is the Tridectin Efficacy Portal. It does a very similar job. What are we looking for in a drench? We're looking for at least 95% reduction. Now, to actually do that, we need to know which drenches are working on your place. So a very simple way to do it is to put zip ties on the ear tags of the sheep that you treat with a particular drench. Pick up the samples on the day of treatment. And here's Dave picking up fresh samples out of the yard. And they have to be less than 10 minutes old if you're doing it this technique, but very handy way of collecting samples without having to take individual samples. But on day 14, after treatment, you do need to sample from the rectum, because you need to actually find those sheep with the particular zip tie on their ear tag, know which treatment they were treated with, and take samples from the rectum. Send them off to the lab and they'll collate a worm egg count reduction test similar to this. And you can see that this test was done on merino ewes. They were treated previously with the Zolvix treatment. And on the day of treatment, they were given 1, 2, 3, 4, 5, 6, 7 different treatments to see how they went. And you can see the average of the worm egg count on the day of treatment was 748, and that the majority of those worms were Barber's pole worm. Now, unfortunately, there's only 1% brown stomach worm, the Teledosargia, and that means that there's probably not enough to make a really rigorous call on resistance for that particular species of worm. So we prefer to do the drench tests when there's a good representation of each of the different worms, but you can't always pick it, so you take what you can. Now, the second stage of laboratory testing looked at the worm egg counts in the sheep after treatment, and by collating the larval culture results with the worm egg count results for each of the different treatment groups, we can look at the efficacy, the percentage of the worms killed by each of the different drenches against each of the different worms. And so on this particular property, the brown stomach worm, Teledosargia, all of the drenches kill it. For Trichostrongylus, or the black scour worm, only the BZ is going to fail here. But for Haemonchus, some of those drenches simply don't stack up, and concerningly, even Closantel wasn't 95% effective. And so the beauty of this is that we can use these results to work out what a combination drench would do, and then you can use a combination drench knowing that it's also going to have good efficacy against the worms that are present on your farm. Now, if we're using long-acting moxidectin, which a lot of producers are at the moment, we have to bear in mind that even though it used to work for about 100 days against Barber's pole worm, for example, because of resistance, these days it doesn't. So we crunched the numbers from the last 5 years in our laboratory to see how long after treatment a long-acting moxidectin injection worked. And we found out alarmingly that only 3 weeks or 21 days after treatment, the worm egg counts had gone back up to 100 eggs per gram. Now, 100 eggs per gram is an arbitrary figure, but it is a very useful one, because if you have 100 eggs per gram of resistant worms coming out of your sheep at this time of year, they'll quickly turn into much larger numbers because of the ideal conditions for worm survival. So that's often taken as a cutoff. It means that without a primer, the long-acting injection has only lasted for 3 weeks, and that's simply not long enough, not getting value out of it. However, producers that used a primer plotted all their figures onto a graph, and we found out that it was at least 51 days before the worm egg counts went up above 100 eggs per gram. And also you'll notice that the peak that we see, which is only about 230 eggs per gram, is about half what it was in the sheep that didn't have a primer. And so that means that you get a lot better value out of your long-acting injections if you use a primer. In fact, you get better than 7 weeks protection out of the long-acting injection, which is, I'm sure, what most producers would be expecting, particularly for their susceptible lambing ewes. So bottom line is, if you're going to use a long-acting injection, use a primer. And a primer is any drench given at the time of the injection that kills the worms inside the sheep. That means that the long-acting injection has the pressure taken off it. It just has to stay in the sheep for a long period of time and stop the incoming larvae coming in. And of course, any worms that develop in the sheep while that moxidectin is present in the body are going to be resistant. And so at the end of the payout period, and you can pick when that is by the time the worm egg counts go back up again, then you also use an exit drench or a tail cutter. That's often given at lamb marking or at weaning. Now we'll just move on to drench decision guides, and this is from the Worm Boss program. You can see here's a mob of sheep. The average worm egg count of mobs of sheep in 2022 was 337 eggs per gram. So this is a pretty good mob to start with. They've got a mixture of black scour worm, brown stomach worm, barber's pole worm, and also some smallmouth bow worms, largemouth bow worms. And even though some of them had, reasonably low egg counts, the average was 337. So if we put those numbers into the Worm Boss Drench Decision Guide, now this does vary from region to region as well, so you have to use the one that's appropriate for your region. When there's less than 40% scour worms, that means there's more barber's pole worms, or greater than 40% scour worms means less barber's pole worms. Then the numbers as an indication for when to drench does depend on the body condition score of the sheep and the pasture condition that they're going into. So you can see that there's a bit of a sliding scale there that you can choose your own threshold for when is a good time to drench those sheep. So this one's for greater than 40% scour worms. This one's for greater than 60% barber's pole worms. And you can see because barber's pole worms lay more eggs, the actual indications for drench are higher. Okay, well, look, let's just summarize the techniques we're using to get through spring and protect our sheep from worms. The first one is monitor the sheep using worm egg counts and observe the animals. Use all those tools that are available, your body condition scores, your farmer char, DAG scores, all those things that will help you to monitor how bad the impact is of the worms on the animals. Second one is paddock management. You can make a huge difference by rotating, using cattle, making hay, all those different things. Select your size for immunity using ASBVs for WEX. So the Australian sheep breeding values for particular rams are very good and produce progeny that have low worm egg counts. Use effective drenches or a vaccine and quarantine drench all your new stock. And the only way you know you're using effective drenches is if you do a drench test. And then finally, or number 4, is feed for resilience. You can actually improve the resilience of sheep against worms by improving their feed intake, and that will be a big benefit as well for their body condition score. So probably relates to better reproduction outcomes as well. Okay, well, that's it from me, and I'll hand back to Fiona, who's going to take us through some of the comments and questions.
Thanks, Matt. Fantastic presentation, as always. We have some questions coming through, which is great. I'll just check both the Q&A panel and the chat panel. So put them in the Q&A panel. Now's your chance. We're about to lose him for a whole year, maybe longer. He might get comfortable over in Japan. So ask Matt your questions now. So our first question we have through is, great question, how far ahead do you need to do the primer drench when you're using your long-acting injection, Matt?
Yeah, this is a really good question, and thanks for highlighting this, because in the past everyone's just said, well, do it at the same time. But in reality, if the sheep are on a fairly clean paddock, you can do it up to, 2 or 3 weeks ahead of the long-acting injection. The thing I didn't mention, though, about the long-acting injection is that the closer to lambing you do it, the more of the chemical, the moxidectin, will be excreted in the milk. And so the rule of thumb there is you want to be at least 4 weeks back from lambing when you give that long-acting injection. And that's to make sure that the sheep actually benefit from it and not the, not the lambs. Because if it goes into the milk, it's essentially wasted because the lambs don't have any worms.
Thanks, Matt. Moving to Barbavax. Is Barbavax available for use in alpacas, Matt?
It's an off-label recommendation. There's no really big-scale studies that have shown that it benefits alpacas. And it would be only something that you do, with a prescription from your vet. So I'm afraid, even though alpacas do get Barber's pole worms, there's no real publicly available information about it.
Thanks, Matt. Next question. What drench does not contain mectin, but specifically here a combination drench?
Yeah, you've actually touched on a bit of a problem in the industry, Ed. Given that most of the combination treatments do have a mectin in them, if you want to rotate away from a mectin, the first option would be for Barber's pole areas to use something that's got naphthalphos in it. And naphthalphos, of course, used to be called Ramitin, and now it comes under different names like Polax and Pole Volt and Napoleon. Notice how they're, very clever marketing. They're making use of the Barber's pole worm for the name of the drenches. And if you look on the back of the pack, there are drench mixing instructions so that you can mix them with, for example, a duo combination drench, so that you do still get the benefit of 3 active ingredients. So that's probably the most common one that's used throughout New South Wales, and particularly in the summertime when Barber's pole worms are a problem.
Thanks, Matt. A couple of questions coming through now. What time lapse is required for testing drench efficacy, and does it vary for the active ingredients you're testing for, Matt?
Just run me past that one again, please, Faye.
What time lapse is required for testing drench efficiency? And then does that vary for the different ingredients that you're testing for? My take on that, Matt, is when you're doing your drench efficacy testing for all the different drenches, is it a different time depending on the active ingredient? What's your time phrase there?
Yeah,
and if that's wrong, I think, Paul, you've asked that question. Please feel free to type in again and we'll readdress it.
No, this is actually a good insight into how the different drenches operate. The drenches such as levamisole, StarTech, Zolvix, and white drenches all act pretty quickly, and they're not in the body for a real long period of time. But some of the drenches, like particularly moxidectin, do stay in the body, and they may have an impact on the worms for up to 14 days. And so we don't want to take the samples too quickly, particularly for moxidectin, because they might be still having an effect on the worms. And so in the past, people have taken samples from sheep as early as 7 or 10 days to check the efficacy of a drench. And that's still done in some places in the world. But in Australia, we've sort of settled on 14 as the best fit for when we're testing a lot of different drenches at the same time. And so one downside of testing too late is that, worms that are picked up from the pasture after the sheep's been treated may confound our results. And so we don't want to do that. That's why we don't go later than 14 days. Going earlier than 14 days, we may see an impact of, any of the mectins, but particularly moxidectin, which can suppress egg laying for, quite a period of time. So 14 days, yes, it's probably a compromise, but it is the best compromise across the board at the moment.
Thanks, Matt. Hope that helps there, Paul. Next question's a great question too. We've got some really good questions coming through. When does ewe immunity begin to rebuild? Is it only once lactation has ended, such as at weaning, Matt?
Yeah, there's been some great research done on this in the past where they've monitored ewe worm egg counts every day, and it generally goes up until weaning. And if ewes are weaned early, for example, and you pull the lambs off after 8 weeks, then they start to suppress their own worm egg count straight away. And so it's simply a function of the fact that they've got all the feed to use themselves rather than having to support the lambs on it as well.
Next question, Matt. This is about exposing larvae in the pasture swads. So the person is in southwest Queensland. In a good year, they've got ample pasture and they use cattle to try and eat it, but obviously they start to eat from the top down. And so unless they keep the cattle on the pasture until it's right down to the bottom, will it have any effect on the larval numbers? Or is it better if they use wethers, or even maybe a combination of both?
Yeah, no, this is really, really important. The first thing is cattle still have a big impact, even if they do only graze down, to 10 or 15 centimetres, because by taking off the top of the sward, they're allowing a lot more dry air and sunshine down to ground level. And it's the key thing about larval survival is that microclimate down at ground level where most of them live. But you're right, if you do graze it lower, and if you use wethers, for example, to graze it lower, then you'll have an even bigger impact. So using cattle's good, using wethers may be even better.
Thanks, Matt. Next question, by dosing sheep with a drench that is meant to be 95% effective and putting them into a very clean paddock, aren't you only selecting for a superworm, Matt?
Yeah, this is a good point. And this really does reflect the challenge for sheep producers in balancing that seesaw where you've got productivity on one hand, where you're trying to get as clean a paddock and as clean a worm, clean a sheep as possible, and maximum growth rates. And on the other side is sustainability, where you get refugia, which is untreated worms that remain in the environment and maintain the susceptible genes, for as long as possible. Now, there's always a balance, and every producer has to choose where they'd like to be on that balance. And so I'm afraid as extension agents, we can put up the options there, but everyone has to work out what their priorities are, whether it's actually having sustainability and having, very, very long use of the same drenches, for example, or whether they want maximum productivity out of their farm. And I think most producers are coming to a pretty comfortable position where they can say, well, we're not going to flog the drenches. We're not going to just put sheep into a clean paddock and only selecting for super worms. We're going to, do something like make sure there's a few worms in that paddock before we put the sheep back in that will dilute the resistant worms and help them to maintain the susceptibility.
Thanks, Matt. We still have quite a few questions, so thanks everyone for staying online. We'll just go a bit over today because they're fantastic questions. I appreciate you all getting involved. Next question is, this person has sheep with some diarrhea. The farmer char was 3. Our worm egg count was less than 200 eggs per gram. The pasture is very lush. It doesn't say what region they're from, Matt, but can the diarrhea be a cause of just the lush grass?
Yeah, absolutely. And this may be a good case study for how, you know, very high moisture content grass on its own will cause, you know, dagginess and you know, loose feces in sheep. And, you know, for example, capeweed, it's like lettuce, you know, it becomes, you know, 90+% moisture. And if you put sheep on that, then they will have very runny diarrhea without any, you know, appreciable worm egg counts. The other important thing about this question is that it sort of highlights the fact that for farmer char, we're checking specifically for Barber's poleworm. Barber's poleworm tends to dehydrate the sheep and make them less susceptible to diarrhea. So we don't often see Barber's poleworm and diarrhea at the same time. And so even at low worm egg counts, we can have reasonable numbers of scour worms, and they're the ones that will be causing the scours and the dagginess.
Next question, Matt. Can you put sheep back into the same paddock after you've just drenched them?
Yes, you can. However, you may be minimising the actual benefit of drenching them, particularly if the same paddock is badly contaminated. They'll just pick up worms again. So that might be okay for a day or two as a way of offsetting the selection for resistance, but then you probably want those sheep, you know, back onto a clean paddock so that they can benefit from not picking up too many worms.
Matt, this person has very high extreme pasture contamination, high rainfall, eastern southern tablelands, and has resistance problems to quite a few drenches by the looks of things. So resistance to tridectin. Is there any other combinations available that they could use? Do you need me to repeat that one?
No, that's fine. I think I've got that. Sounds like this is a bit of a problem that would need to have some more specific testing done, in particular the worm types on that place, because different combinations work well against different worm types. But I'd be definitely finding out if you had Barber's pole worm there, given that it's the southern New South Wales area. You may or may not, and be making a drench decision based on whether you had Barber's pole or not. But the newer drenches in general tend to work better.
Thanks, Matt. And I think this is almost my last question. Oh no, got a few more. How do I start a Barbavax program in a herd that's never been vaccinated? They are under the understanding to start only with young stock, about 12 months of age, and the rams. And would this be effective to build resilience in the flock over time?
Yeah, great question. So the Barbavax product details are outlined on the label. The easiest way to actually get into Barbavax is to vaccinate lambs. And the best time to do it is the first time at lamb marking. So when they're quite young, that's usually between 2 and 8 weeks of age. Second time, 3 weeks later. And then the third time usually is at weaning. So in the first 6 weeks, and sorry, Fiona, I know this is your area of expertise, not mine, but in the first 6 weeks, you've given those lambs 3 injections, and they now have immunity against Barber's pole worm. Now, it's not 100%, it's not complete immunity, but it will definitely suppress both the numbers of worms inside the lambs and also the number of eggs that those worms are putting back onto the pasture. So it's a big benefit to start off with your lambs. Once they're vaccinated, you can then repeat vaccinations later on because the vaccine only lasts about 6 weeks. And so if you continue to have bad pressure from the Barber's pole worms, you can continue to vaccinate them 6 weeks after that. And then the next group, of course, are the ewes, and you can vaccinate ewes as well, but the same principles apply. They don't get the protection until after the 3rd vaccination. Hope that helps.
Thanks, Matt. Just going back to our question about triple actives.
Mm-hmm.
So the resistance problem they're having is with scour worms and not Barber's pole worm. And they're worried about using single actives if that will lead to having no options ahead down the track.
Yeah, really good point. The emerging worm that we've seen over the last 5 years is the brown stomach worm, and there's a lot of them that are resistant to triple active. So you're definitely not alone there. Generally, the new drenches, Zolvix and StarTech, are working quite well against those brown stomach worms. So that would be the sort of very broad suggestion. And sorry, I can't be more specific there.
All good. And a great question to finish on. It always comes up, Matt, and it's a follow-on from drenching and putting them back in the paddock. How long after you drench should you move them onto that clean paddock? Immediately, 24 hours later, 3 days later, a week later? What is it, Matt?
Yeah, no, this is a great question. It does change according to how wormy the paddock is, and also what the producer is trying to achieve. And so there isn't a single answer for this. It really does depend on whether you're trying to go for refugia, sustainability, lower selection for resistance in the worms, or if you are running a big place and you just want productivity without having to do much mustering, then the default might be, well, I know that paddock that I'm putting them into has already got some susceptible worms on it, so I'm going to just drench and put them into the new paddock straight away. However, all of those strategies, the 24-hour strategy, 3-day strategy, will work. The second part of that question, which relates to contamination of paddocks, is that generally the passage of digester through the gastrointestinal tract of the sheep takes about 3 to 4 days, but if they're on really dry feed, it may take up to 7 to 8 days. And so if you're drenching them and hoping to avoid contaminating the new paddock, then you do want to keep them, in the same paddock for, the period of time that represents, that passage of digester. So usually about 3 to 4 days.
Great, thank you, Matt. That brings us to the end of the webinar for today. And I'd like to once again thank Matt for joining us, and thank you for your commitment to AWI Extension New South Wales for many years now. We will miss you tremendously, but we're looking forward to your return and hope that you gather lots of wonderful information for you to share with us when you get back. So thank you, Matt.